Alright guys, exams are on the back table. They are in alphabetical order. If you don't pick them up before class is over, I'll just shred them after class. I can't handle having like 1,200 exams on my desk at the end of the semester, so I don't keep them past the opportunity for you to come and pick them up, unless that you know you're not going to be in class. And you send me an email letting me know about that. So if you want your exam, make sure that you go and pick it up before the end of class. After that, I'm just going to get rid of them. Also, we are behind. You guys ask a lot of good questions. I was telling my husband that this is like the best 227 class I've ever had a couple of weeks ago. So you guys ask a lot of really good questions, and that's fantastic. But that combined with me being sick has made us really far behind. So we are going to skip the rest of chemistry. The end of the chapter looked at, yeah, you're also disappointed. The end of the chapter looked at organic molecules, and some organic molecules that are important to the body: carbohydrates, proteins, fats, and nucleic acids. We're just going to skip that. It's something that we can live without at this point in time. So it won't be on your exams. I don't expect you to go learn it on your own. It's just something that you can cross out of your notes, and that we're going to skip over because we've got to get into some biology in this class. And so we're going to go ahead and move into cells, and even still, we're probably kind of a little bit behind. But we're going to start with cells today, and hopefully get caught up somewhat to where you guys are in lab. I think you're doing skin in lab this week, so we're kind of behind. So what we're doing is we're kind of building again our complexity and our background. So we've talked a little bit about what life is, how it's maintained, some of the characteristics of life in chapter 1. And then we started out with kind of the simplest building blocks that we have in the human body, and looked at atoms and their components, and looked at chemicals and how atoms come together to form molecules. And we also talked about some basic chemistry that is necessary for you to have an understanding of to continue on through 227 and 228, and that's basically equilibrium reactions and pH. So the rest of that would be nice stuff to have, but I'll introduce it as we come upon it in 228, and we'll go ahead and skip the rest of the organic molecules and just go ahead and get started with cells. So if you remember that one slide that we had up first or second day of class, where we had kind of the order of life, we talked about how chemicals organize themselves into cells, and cells organize themselves into tissues. And so this particular chapter is now getting us back into the realm of the living, where we're talking about cells, those basic building blocks of life. So we are all composed of about 70 trillion cells. That's a 70 with 12 zeros after it, just to put that in perspective for you. But these are the basic unit of life and what our bodies are composed of, essentially. So I want to give you some general information just about cells. First of all, in our bodies, it's estimated that there are about 260 different types of cells. So muscle cells, skin cells, cardiac muscle cells, nervous tissue cells, many, many different types, about 260. Cells vary quite a bit in their size and quite a bit in their shape, as we're going to be talking about as we move through this particular chapter. So the smallest cell out there is about 3 microns in diameter. Any idea what the smallest human cell is? Nobody's willing to even take a stab? You guys talk more than any class I've had. Come on.
Are you ready for?
Oh, jeez. Okay. Fine, I'll say it. The smallest cell in the human body is a sperm cell. It's about 3 microns. Okay. The largest cell in the human body is about 35 microns in diameter, so significantly larger than that. It's just visible at that size to the naked eye if you have very, very good eyesight. Any idea what the largest cell in the human body is? It's the egg. Okay. Yeah, it's the egg. So a large kind of discrepancy in size, with the sperm being at the smallest end, the egg being at the largest end. And as I mentioned before, they also vary quite a bit in their shape. So this goes back to the idea that we introduced in chapter 1, kind of one of the hallmarks of anatomy and physiology, and really biology in general. And that is the idea that form fits function. Okay, and we have cells of different shapes and different sizes because their functions vary. And so an egg cell needs to be very differently shaped from a sperm cell. They don't have the same function. A sperm, of course, has a long whip-like tail, and that's going to allow it to propel itself through the female reproductive tract. Just to give you an example of some of the differences in shape and size that we have for cells in the human body, I've put some pictures up here. I want to talk first of all about red blood cells. And if you look closely at the shape of a red blood cell, if you view it from the side, it has what's known as a biconcave disc shape. What that means essentially, shoot, okay, totally ignore that. Here we go again. All right, it has a shape, something like this. Okay, so it's concave on both sides. And this particular shape makes a red blood cell capable of doing what it needs to do. Okay? So what's the function of a red blood cell? What's the major thing that it does for the body? Carries oxygen. So it picks up oxygen at the lungs, and then it moves through the bloodstream to all the cells, tissues of the body, and it unloads that oxygen. Well, red blood cells, when they get into blood vessels, which are known as capillaries, these are the smallest capillaries in the body. Capillaries are actually so small that these red blood cells have to move through single file. Okay? It's just wide enough for a red blood cell to squeeze through. And even that, moving through single file, really isn't enough. The red blood cells still have to kind of twist and contort themselves and bend to even be able to fit through the capillaries. And having this biconcave disc shape makes a red blood cell very, very flexible so that it can pass through the 60,000 miles of blood vessels that we all have in our body and have a lifespan of 100, 120 days before it starts to break down and starts to become destroyed by this process of trying to squeeze through these teeny tiny blood vessels all the time. Yeah, it just makes it really flexible. What's that? 100 to 120 days. Not something you need to know. I just want you to know it's flexible, and I'm just giving examples of how. The other thing that this particular shape does is it sets up a situation where if you look at this cell, we have a very high surface area to volume ratio. So another way of saying that is there is no part of the inside of the cell that's very far from the outside of the cell. And the reason that's important, this particular shape, is we need this thing to carry oxygen. So oxygen's going to move in, okay, and it's easy for it to get into the center of the cell, but it's very quick for it to move out. If you think about a cell, okay, that was shaped like a globe, a circular cell, it would take a lot longer for oxygen to get into the center of the cell and a lot longer for oxygen to get out. This makes it much, much better at binding to oxygen and releasing oxygen to the cells and tissues of the body. And that's why we see this particular shape with red blood cells, because again, we have this idea that the form, the shape, the structure is going to be matched up with the function, and that shape is going to allow it to best perform its particular role in the body. So here we have another cell, this one that's represented here in pink. This is actually my favorite type of cell in the body. They're fascinating. This is what's known as a dendritic cell. And a dendritic cell is a cell that works in the immune system. And if you look at this cell, you'll notice that we've got kind of the normal, typical, circular, globular cell that we're used to seeing if you've had some basic biology. But you'll also notice that it has these arms, okay, as we've called them before, or pseudopodia, that extend off of it. And we actually talked a little bit about this type of cell very, very early in the class. And I had you guys kind of look at this and use your developing scientific minds to say, why would these things be here? This isn't something that we see on normal cells. And you guys came up with some ideas, and one of the things that you said is their arms and they're used for grabbing things. And that is exactly true. What these dendritic cells do in our bodies is they are circulating through our bodies all the time, and they are looking for things that have been introduced into the body that should not be there. Okay, so they're kind of like the policemen or the security guards of the body, if you will. And when they find something that they recognize as foreign to you, that they recognize as something that should not be there, they actually grab a hold of that thing, okay, whatever it is. Maybe it's a virus, maybe it's a bacteria, maybe it's a fungi, Maybe it's a protein that has been introduced that they're detecting as not a part of you, something that shouldn't be there. And they take that harmful foreign thing and they show it to another cell. This is a lymphocyte, okay? Lymphocytes are kind of like the generals of our immune system. So we've got our security guard or our policeman, our dendritic cell. He's found this harmful thing. He's taken it to a lymphocyte saying basically, look what I found. Okay, this should not be here. And you're kind of the general of the immune system, if you will. You, lymphocyte, need to go tell all of the other immune system cells what I found. You need to let them know what it looks like so that they can now go out en masse and start looking for this particular thing, because if there's one, there's probably more, and working to destroy it before it has an opportunity to make the body sick, essentially. So amazing cells, amazing interaction that goes on. We'll talk about that for like a day when we get into 228, this interaction with the immune system. But again, they wouldn't be able to do this if they didn't have this special structure to them. So their form allows them to perform their particular role, their particular function in the body. If you look at this cell here, this is what's known as a neuron, or also just more commonly called a nerve cell. So these are the types of cells that we find predominantly in the brain, also in the spinal cord. And you'll notice that we've got this little cell right here, nucleus in the center. And then what's kind of different about this cell from the typical cells that you're used to seeing in a basic biology class is it has these very, very long extensions that run away from it. These extensions can be up to 1 meter long, and what they do is they allow for communication to be sent from this cell to another cell, which is located a very long distance away in the body. So these cells are actually capable of generating electricity and sending an electrical current, okay, down these extensions, much like electricity running through a power cord to communicate with some other part of the body. If you think about all the movements that you do, like me flexing my arm, I had to plan that movement in my head. I had to generate an electrical current with neurons in my head, and that electrical current then traveled down extensions from neurons in the brain to tell this muscle to contract. I mentioned that these extensions can be up to 1 meter long. So we do have extensions that extend from the brain all the way down the spinal cord, down to the sacral area in the buttock. So those would be the longest ones that we have in the human body. And if you're very tall, maybe up to about 1 meter long. If you think about animals like giraffes, okay, that have this very long neck, they're going to have neurons that start in the brain that send extensions all the way down their neck, down to the appendages and the other parts of their body, so that the brain can control those particular organs and those particular structures. And in giraffes, we actually see these extensions being up to about 15 meters long, believe it or not. So very, very long cells. They have these extensions, this particular shape to them, to allow them to generate and transmit these electrical signals so that they can control what's going on in the body.
So I guess my question is, you know how you have like a knee-jerk reaction when somebody hits your knee and stuff like that? It doesn't go all the way back to your brain?
That particular one doesn't. So a lot of reflexive things that are meant to protect the body are done in the spinal cord, and that in particular is one that's meant to protect the body that is coordinated in the spinal cord rather than going all the way to the brain. So some do, some don't.
Does it go back to like the nucleus of the neuron itself, or is there some other process?
It does. So usually what happens with that particular one is you have a neuron that's sending a signal up into the spinal cord. That neuron synapses with another neuron in the spinal cord that comes right back down and causes that contraction of the leg. So that's something that we'll get into a lot when we get into Unit 3, and it'll mostly be something that we talk about in lab. But some do go all the way to the brain. They take longer when they do because they're traveling a longer distance, and things that need to be protective or are meant to protect the body from harm usually are just controlled in the spinal cord. All right. So we've had kind of a basic introduction to cells. We've talked a little bit about their purpose in the body and the fact that this idea of form fits function is true on a whole body level, but it's true on a cellular level as well. And so what I want to do now is do some very, very basic biology, talk just about the 3 major parts of a cell, and then what we'll do after this is we'll come back and we'll talk more specifically about cell membranes. So we'll probably spend all the rest of today and possibly Thursday as well talking just about cell membranes. Okay? And then we'll talk a little bit about the cytoplasm and a little bit about the nucleus. But for the standpoint of this class, what's important that you have an understanding of is actually the cell membrane, okay? Because it is, to a large extent, determining how cells work, okay, and how tissues and organs work based on the properties of the cells that make them up, essentially. So the cell membrane is basically, you could think of it like the skin of the cell. It's this structure that surrounds the cell, that separates what's inside the cell from what's outside the cell, that allows the cell to keep those two things distinct, allows the cell to maintain boundaries, essentially. The cell membrane is also commonly referred to as the plasma membrane. And the reason for that is the nucleus is also surrounded by a membrane, so it's a membrane within the cell. And if we call this plasma membrane and this nuclear membrane, it just makes it a little bit more clear about what particular cell membrane we're referring to. So sometimes you'll hear me use cell membrane, but more specifically plasma membrane would be the distinguishing term for this particular boundary. The other thing that I want you to know, and I'm just going to mention this now, and then we'll come back and I'm going to draw this out for you and talk about the very, very important properties that this gives to the cell. But the cell membrane or the plasma membrane is composed of what's known as a phospholipid bilayer. So a little bit about phospholipids. They are a type of fat. Okay? So the cell membrane itself is mostly fat. It has kind of the consistency of olive oil, okay, because it's mostly fat. Olive oil is a fat. What that means is the cell membrane is not tremendously rigid. I threw away my son's soap bubbles, but usually I bring them in and I blow soap bubbles in class while I'm talking about this, because it's very similar to the consistency of a soap bubble. So if you think about, okay, blowing bubbles with a child or soap bubbles, you blow them, they're these circular things, but that membrane of the soap bubble kind of twists and turns on itself, and it's not very rigid. You can actually poke it by putting your finger through it. And that's the same thing with a cell membrane, okay? They are not stable, rigid structures. They are not like that at all. They're more like a soap bubble. And so what we actually see is embedded in that cell membrane, we see some things that are going to help to stabilize it and help to make it a little bit more rigid. And we'll talk about those things a little bit later on. So more about the phospholipid bilayer. I'm going to actually draw that out for you so you can see it and understand how it works and how it helps the cell to some extent to be able to maintain its boundaries and be able to keep its internal environment distinct from its external environment. Second major component of the cell is what's known as the cytoplasm. So the cytoplasm is basically everything that's inside the plasma membrane but outside the nucleus. So it contains all this stuff in these areas where I'm kind of running my hand along here. The cytoplasm has two components to it. Cytosol is a jelly-like fluid, okay, so it's mostly water. The internal environment of a cell is this mostly water, jelly-like fluid, which is known as cytosol. Suspended in the cytosol, okay, we have little structures, some of which you can see on here, which are known as organelles, okay. So organelles are those structures that you guys learned in lab last week, such as the mitochondria, such as the endoplasmic reticulum, such as the Golgi apparatus. They're just structures that are embedded in the cytosol, that perform specific functions for the cell, that allow the cell to do all of the things that it needs to do. So we'll talk a little bit more about each of these organelles and their particular function in the cell as we continue through this chapter. The last thing that I want to mention, and just mention very briefly, is the nucleus. I mentioned that the nucleus has a nuclear envelope or a nuclear membrane that walls it off, that separates it from the rest of the cell. It is within the nucleus that we find 99% of the cell's DNA. So there's about 1% actually in the mitochondria, but most of it is within the nucleus itself. And when a cell is not actively dividing, Okay, you guys may have learned this term in lab last week. Chromatin is DNA mixed with proteins in a less condensed form, essentially. When a cell gets ready to divide, you know that the DNA in the nucleus starts to condense down. It starts to organize itself into what are known as chromosomes. And you guys would have had a chance to see some of that going on with the slides that you looked at in lab last week. But when you've got a cell that's not in the process of dividing, okay, and that DNA is in a less condensed form, it's not picking up stain, so the cell that's in interphase, that DNA mixed with proteins is referred to as chromatin. So very, very basic introduction to the major parts of a cell. And as I promised, what we're going to do now is come back and talk, probably for the rest of today and into Thursday, just about the cell membrane. And then we'll talk a little bit more about these structures that are contained within the cytoplasm as well, so that you've had a basic introduction. And now we're going to kind of focus down on the cell and its major parts for a while. So before we continue, anything that I could explain better? All right. If not, okay, again, we're focusing now on this structure, the cell membrane, and I want to start by drawing you a picture. We talked about a little bit ago that the cell membrane is composed mostly of fats, which are known as phospholipids. And these phospholipids actually form a bilayer within the cell. So if you can imagine this right here as a cell in your body, here's the nucleus in the center. If we were to take just a little part, okay, of the cell membrane or of the plasma membrane and look at it, okay, under a high-powered microscope, what we would see is something that looks like this. Okay, we've got these little fat molecules. Believe it or not, they do have kind of this particular shape that I'm drawing to them. It looks like a sucker with two sticks, basically. And these fat molecules, or these phospholipids, are arranged in the cell membrane, okay, like this. They're actually forming a double layer. So that's why we refer to the plasma membrane or the cell membrane as a phospholipid bilayer. We've got two layers of phospholipids essentially that are making it up. Now there's something about the chemical properties of these phospholipids that is very, very important to understanding how the cell membrane allows the cell to kind of maintain its boundaries and keep its internal environment distinct from its external environment. And that is, okay, this circular portion of the phospholipids are the heads, and the heads actually have a charge to them. If you were to look at the molecule, the head of this molecule is charged, okay? So we've got heads that are facing out from the cell. We've got heads that are facing internally into the cell. These heads are charged, and because they are charged, they are what we refer to as hydrophilic. So if you've had some med term, what does hydrophilic mean? Water loving, okay? So they're charged, which means that they are like water. You'll remember that water is a charged molecule, and we talked about the reasons that that is. And so they like other molecules that are charged, and we refer to these heads as being hydrophilic, okay? What that means essentially is these heads want to be in a watery environment. And that's fantastic for these heads that are facing into the cell, because what do we have going on in the cell? The cytosol, right? Which is like almost 100% water with some things that are suspended in it. So we've got this watery environment inside the cell. Anybody know what the interstitial fluid is? It is a fluid that bathes basically all the cells of our body. So what we've got on the inside of cells is this watery environment. We're like 71% water. So what we've got on the outside of our cells is they're bathed in water. We've got this watery environment again. And that's just great because we've got the heads that are facing out into a watery environment, and that's what they want. We've got heads that are facing into a watery environment, and that's what they want as well. So these heads are very, very happy about that. The tail portion is referred to as hydrophobic, okay? And the reason that the tails are hydrophobic. Can anyone see that? I've never tried to use yellow. Okay, the reason that these tails are hydrophobic is because they are not charged like water. So if you break down the term hydrophobic, if you've had some medical terminology, what does that mean? Okay, they fear water, they don't want to be in a watery environment, and that sets up a situation essentially where water is happy to be here with the heads on the inside. It's happy to be outside. Anything that's charged, okay, ions, those types of things that might be dissolved in the water, they're happy to be there associating with these hydrophilic heads. But if water tries to squeeze in between the phospholipids, it's not going to. It's going to move back out because we've got this water-hating environment, okay? If you have ever tried to mix oil and vinegar, you know that it's very difficult. They will separate back out. They won't mix, and the reason for that is vinegar is hydrophilic, okay? Oil is hydrophobic, and so it's the same idea that you won't have these things mixing. And so that sets up a situation where water, okay, things that are charged on the inside are not going to pass out because they cannot pass through that hydrophobic barrier that we have all the way around the cell. And things that are on the inside, okay, are not going to pass in, or on the outside, are not going to pass in because they can't pass through that hydrophobic environment. So that's going to allow us to keep, for the most part, what's on the inside of the cell inside, and what's on the outside of the cell outside, separate and distinct from the cell. Okay? So water, water-soluble substances, things that are charged like water, things that can dissolve in water, they cannot pass through that barrier because of the orientation of our phospholipids. The only thing that can pass are very small fat-soluble substances, okay? Because remember, fats are not charged, they're hydrophobic, and something that's fat-soluble is not going to have a problem passing through this hydrophobic environment from the inside to the outside or from the outside to the inside, okay? There are really only 2 molecules of any importance that we're going to talk about in this class that can pass through the cell membrane unimpeded because they're very small and because they don't mind this hydrophobic environment. One of these is oxygen. And the other one is carbon dioxide. Okay, and if you think about those 2 molecules, that's really important that they can pass unimpeded, right? Because what is oxygen doing in the body? Once it's picked up by red blood cells and it's transported to a cell, what's that oxygen used for? Okay, cellular respiration. It's used to produce energy, essentially. And that reaction where energy is being produced happens inside the cell. So we want oxygen to be able to pass unimpeded inside the cell. What's a waste product of that cellular respiration or that reaction where we make energy? Carbon dioxide, okay? We don't want carbon dioxide building up in here. It would be very toxic to the cell. So carbon dioxide is really the only other important one that we're going to talk about that can pass unimpeded. And it's going to pass out of the cell into the blood and then be transported back to the lungs so that we can exhale it. Okay? So that's a little bit about this particular membrane and how it kind of functions to keep the inside environment of a cell separate and distinct from that outside environment. I know it's a little bit complicated, it might be a little bit abstract since it's not something that we can see and put our hands on. Is there anything that I could explain better about that before we build on it and we continue on with this particular topic? Okay. So I talked a little bit previously about the fact that cell membranes are made of fats. Okay? So they have the consistency of olive oil essentially, or maybe of soap bubbles if you can picture those in your mind. And I also mentioned that we have certain substances that are embedded in the phospholipids for different reasons. So what I want to do now is talk about some of those substances that are embedded in the cell membrane. One of them is another fat, which is known as cholesterol. Cholesterol is a much more rigid molecule than a phospholipid. And so if you look at this picture here, we've got a portion of a cell membrane. Here's the inside of our cell, the outside of our cell, here's our phospholipid bilayer, and you can see how those particular chemicals are arranged. But you'll notice every so often we have a molecule of cholesterol that is embedded in that cell membrane. That's going to help to anchor the cell membrane to some extent, to make it a little bit more rigid, because cholesterol is a rather rigid molecule. It's also going to help to make the cell impermeable to water. Okay, again, we've talked about water not crossing, but cholesterol is also a fat. It is hydrophobic, and that's going to help to prevent water from moving from the inside out, from the outside in, okay, just by squeezing through or between adjacent phospholipids, essentially. The other thing that we have embedded in the cell membrane are proteins. So there are a couple of different types of these, and I want to talk about them generally, and then we'll talk about some specific examples. But if you look at, again, the cell membrane, we've got our phospholipids. You'll notice that there are some additional structures represented in purple and green that are embedded within the cell membrane. So we've got what are known as integral proteins. Integral proteins essentially are proteins that span the entire cell membrane from one side to the other. Peripheral proteins are proteins that are just attached either to the outside of the cell membrane, or they're attached to the inside of the cell membrane, but the protein itself doesn't span across the entire membrane. So we've got integral and we've got peripheral. And what I want to do next is talk about some of the specific functions of peripheral proteins and some of the specific functions of integral proteins as well. So we're going to start out with peripheral proteins, and you'll remember that these are those proteins that are attached either just to the outside or to the inside. They don't span across the entire membrane itself. And one example of these are proteins which are known as CAMs. So CAM stands for cell adhesion molecule, and there's several different reasons that we would find these cell adhesion molecules or these CAMs attached to the outsides of cells. The first thing is these proteins that are on the periphery, that are extending out from the outside of the cell, allow cells to touch or bind. Okay, when we start getting into tissues here, you've looked at some tissues last week in lab. What holds the cells together in those tissues, which keeps them kind of aggregated together, are the CAMs, the peripheral proteins, actually attaching them and holding them to other cells. I want to give you another example of this, of peripheral proteins allowing cells to touch or bind. If you look at HIV, the virus that causes AIDS, it has a peripheral protein on the outside of its cell wall. I guess it's a virus, so it doesn't really have a cell wall. But it does have a peripheral protein. And the peripheral protein kind of surrounds it. There's thousands of them that are attached to that outer wall. And this peripheral protein kind of looks like how I'm drawing, and it has a name. The name of this peripheral protein is GP120. And HIV, when it's introduced into the body, uses this peripheral protein to attach to our immune system cells and open those cells up so that HIV can get into the cell itself. So if you were to look at an immune system cell of the body, okay, immune system cells have their own peripheral protein, which is known as CD4. So this is my representation of CD4. And what happens when HIV in the body encounters an immune system cell is GP120 binds to CD4. Okay? There's then a second peripheral protein that most of us have on the outside of our immune system cells. The second peripheral protein is known as CCR5. After binding CD4, GP120 binds to CCR5. That opens up the cell and allows HIV to put its genetic information into the cell and to infect that cell essentially. About 1% of Americans do not have CCR5, and because they do not have CCR5 on the outside, of their immune system cells, they are completely immune to HIV. It is something that cannot be passed to them. So they can be exposed to it over and over and over again, but there is no way for HIV to get into their cells, to take over their DNA and their genetic equipment, and to create an HIV infection because they're just simply missing that particular protein. Yeah?
Isn't it the same with red blood cells? Like type A, type AB, like they have that protein on the outside?
They do. So they have different proteins on the outside that distinguish what blood type you have, basically. So if you have type A, you have the A protein on the outside. If you have type B, you have the B protein on the outside. If it's AB, you've got both proteins on the outside. Type O doesn't have any of them. So the only other thing that I wanted to say about this with regards to CAMs, these peripheral proteins allowing cells to touch and bind, is one of the things that happens in cancer is this process fails. Okay, so cells become abnormal. The process that would normally hold cells together in a tissue fails, and you get cells that start to break away from their original site, get into the lymphatic system, get into the bloodstream, travel to organs or body tissue somewhere else, and cause cancer there in that particular area. So this is one of the things that leads to cancer spreading, is these peripheral proteins failing, okay, in cancerous cells, and no longer binding those cells together so that they can metastasize or they can spread throughout the body, essentially. So very, very important, the roles of these CAMs in allowing cells to touch or bind. They can also help cells to migrate, so they can act as arms, okay, which cells can use to kind of move their ways from the body. They can actually, believe it or not, send SOS signals. So we've talked about, okay, a virus getting into a cell. One of the things that happens when a virus gets into a cell is a cell will actually create a peripheral protein that says, help, I'm infected, immune system, come and destroy me because I'm infected with a virus. This virus has taken me over, and if you don't destroy me, I'm going to cause an infection that spreads to the entire rest of the body, essentially. So really some amazing things that they can do with regards to that, again, that we'll talk about in the immune system in 228, and being able to send signals and communicate with other cells to let them know essentially their health status. We can see them acting as mechanical sensors and then just transmitting signals to cells in other ways as well about their nutrient levels or whatnot, so that there is this communication that's going on between all the cells of our body, essentially. So that's a little bit about these peripheral proteins, again, proteins that are extending from the outside of the cell membrane, okay, and not necessarily spanning entirely the cell membrane. I wanted to talk just a little bit also about the integral proteins. So these are the ones that are embedded in the cell membrane. They completely span that cell membrane from one side to the other. And there are several different types of these that help the cell to perform its particular roles in the body. So the first of these that I want to mention are what are known as aquaporins. Pores, basically, is another term for them. And if you could imagine this little integral protein here, and it had a little opening or a little channel that went all the way through it from one side to the other, that would be the basic structure of an aquaporin or a pore. What these particular channels allow for, or what these integral proteins allow for, is water to enter or exit the cell, okay, under certain conditions. So I want you to think about this for a moment and relate it back to what we've talked about previously. Why, okay, for water to enter or exit the cell, do we need to embed a protein in the cell membrane with a channel through it? Okay, so I'll give you guys a moment to think about that. Talk to your neighbor if you'd like. But what's going on there? Why is that a necessity? All right, so let's bring this back together. You guys have had a little bit of time to think, a little bit of a break, maybe talk it over. Why does water, because under some circumstances we do need water to leave the cell, we may need water to enter the cell. Why does it need to travel through this protein? Okay, it cannot, so a lot of things, carbon dioxide and oxygen, they can just squeeze through these adjacent phospholipids, kind of muscle their way through, okay, on their way in or on their way out, and they're not bothered by this hydrophobic environment. Water is, okay, water would never cross through here, just like vinegar would never cross through a layer of oil, essentially. And so what we've got to do is we've got to embed something in this hydrophobic area, okay, that makes it possible for water to be able to cross through it without being exposed to the hydrophobic area. And that's why we have these aquaporons, or these pores, that are embedded in cell membranes. And if you were to see a typical cell membrane, there would be thousands, okay, of these channels that are just specifically for water actually embedded in it so that we can allow water to cross. Okay? But here's the thing, this gives the cell control over water moving to some extent, because it can open the channels or close the channels to allow for water to pass or not to pass when it needs it to. So we've talked about aquaporons. Channels, the next thing on the list, same idea as an aquaporon, except for they are specific for ions. Okay? So why would ions need a channel? Why would they need a protein embedded in the cell membrane, pull all the way through the center of it to be able to cross? What is it about ions that would prevent them from moving into that hydrophobic environment? They're charged, right? And because they're charged, they are hydrophilic. They are like water, they would dissolve in water, and because they're hydrophilic, an ion, even though it's teeny tiny small, is not going to cross through here. It's not going to squeeze through these adjacent phospholipids in either direction, because it doesn't want to be in that hydrophobic environment. It's not going to move into the hydrophobic environment. There are lots of situations in the body where we need ions to move across the cell membrane. Muscle contraction is an example of one that we'll talk about in chapter 9. And so we've got to have a way to allow these ions to move, and that's going to happen through, again, thousands of channels that are embedded in the cell membrane. But again, because they're channels, we can open the channels to allow ions to cross. We can close the channels to prevent ions from crossing. And that's going to give the cell some control over what exits, what enters it, essentially. Here's a couple of other things that I want to mention just briefly that you'll find embedded in the cell membrane. Receptors, I'll give you kind of a formal definition, and then I'll give you an example that maybe hopefully will make it not so abstract. Receptors bind to incoming molecules, triggering responses within the cell. Okay? So again, kind of abstract. You get something that's binding here on the outside of the cell, and that triggers a response that happens on the inside of the cell. So here is a real-world example of this that many of you may be familiar with. Okay? What is the hormone that's released in our bodies when blood sugar starts to get high?
Or insulin.
Okay, it's insulin. Okay, so many of you may be familiar. We eat, okay, we break down carbohydrates, they get taken up as sugar into the blood, and after eating, therefore, our blood sugar starts to get a little bit high. Well, blood sugar can't get too high, because if it does, it starts to make the blood syrupy, it thickens it, and that's very damaging to the organs, and it makes it hard for the heart to pump and all those things that you could imagine. So what our body does after we eat and that sugar starts to get into the blood and blood sugar starts to increase is we release a chemical, a hormone, which is known as insulin. And what insulin does is it binds to receptors on the outside of the cell, and it causes the cell to start taking up sugar. Okay? So the cell is taking that sugar from the blood, and it's going to use that sugar to make energy through cellular respiration, essentially. It's glucose that it's taking up. So that's a good example of an incoming molecule, insulin, triggering a response, the take-up of sugar on the inside of the cell, essentially. And we've got lots and lots of those receptors, thousands of those receptors embedded in the cell membrane. The last thing is what's known as a transport protein. A transport protein is kind of the same idea as a channel, essentially, but it's going to be transporting molecules that are very, very large. So things that are so large that they can't diffuse, okay, and that's a term that you should know because I'm going to be using it quite a bit from here on out. Diffuse means just squeeze in between adjacent phospholipids, move through that hydrophobic environment to one side or the other. If they're too big that they can't diffuse and they can't fit through a channel meant for an ion, ions are very small, or a channel meant for water, water is very small. Then we have these transport proteins that are embedded in the cell membrane that can allow these very, very large things to enter or very, very large things to exit. So same idea basically as a channel, but specifically for large things that can't fit through channels is the idea behind the transport protein. So I want to introduce a term to you here, and this is actually something that we've been talking about all day ever since I mentioned the idea of cell membranes, but I want to give it kind of a formal definition, and then from that build on that to help you understand what's going on with cells as far as their plasma membranes allowing them to maintain boundaries. So we have kind of established the idea with this diagram over here and the way that phospholipids are oriented in the cell membrane, that the structure of the cell membrane allows it to control the entrance and exit of some substances, okay? So water is just not able to cross because the cell membrane has that hydrophobic barrier. And so the only way that water can cross is if it is moving through a pore and the cell opens up that pore. Ions, okay, are not able to just freely cross because they're charged. They're not going to pass through that hydrophobic environment. So the only way that they can move in or move out is for the cell to open up a channel and allow them to do so, okay? So some things can't cross freely, some things can. Carbon dioxide and oxygen we've talked about because they are very small. They can squeeze through those phospholipids because they are hydrophobic, they're fat soluble, they don't mind that hydrophobic environment that we have here by the tails of the phospholipids. But the cell's got some control essentially over what enters, what exits. It doesn't have total control, but it has some control over what's able to cross its cell membrane. And this idea is known as selective permeability. So some things are permeable, meaning they can cross the cell membrane. Other things the cell membrane is not permeable to, meaning that it does not allow those things to cross. And there's some selection in it, okay? So these things can cross, these things can't. Sometimes this may be able to cross an ion or something when the cell allows it to, when the cell opens up a channel for it. So that's kind of the idea of selective permeability. And again, this is getting back at the fact that the cell can regulate, okay, what enters, what exits to some extent, and that allows it to maintain its boundaries, to keep its internal environment distinct from its external environment, which is one of those things that is necessary to be considered living. Okay, so it's meeting that particular characteristic of life. So we have been talking all morning about the idea that cells can regulate to some extent what crosses their cell membrane. What I want to do now is talk about what happens mechanically when something actually does cross the cell membrane. So when a cell allows a substance to pass through its cell membrane, mechanically, how does this actually happen? And so this slide here, again, I'm going to use to define some terms, and then we'll give a whole bunch of examples and give you some thought questions and some activities to help you hopefully understand what's going on in passive transport, okay, when something passes through the cell membrane passively, what's going on in active transport when something moves across the cell membrane through an active process. So I'm going to give you just some definitions here, and then we'll look at some examples to hopefully cement this in your mind a little bit deeper. When a substance passes through the cell membrane, okay, moves through the cell membrane passively, what that means is the substance simply diffuses, okay, so it squeezes through adjacent phospholipids. It diffuses across the cell membrane or through a channel, okay, an ion channel or something like that, from an area of higher concentration to an area of lower concentration. So this is kind of the kicker here, okay, but it's moving down, or what's known as down its concentration gradient. If you have a cell, okay, and there is 80 molecules or 80 ions of sodium on the outside, and there's 1 ion of sodium on the inside, okay, in which direction is sodium going to move based on what's happening in passive transport? It's going to move from the outside in, because it's going to move from its area of higher concentration to its area of lower concentration. One of the things that's unique about passive transport, and this is really kind of the kicker that I want you to understand from this particular slide, is for something to move passively. For those sodium ions outside the cell to move inside the cell, no energy is required. Okay, the cell doesn't have to pump them in. They are just going to do it themselves through some natural processes that we're going to talk about with the next slide. So passive transport, the two keys are a substance moves from its area of higher concentration to its area of lower concentration. And the other key is it doesn't require energy. Okay? ATP is kind of the fuel for our bodies. Gasoline is the fuel for our car. If we're not expending ATP, we're not using any energy essentially to pump those things. They're just moving on their own, again, due to some natural processes that we're going to talk about. Active transport is very different from this. Active transport requires energy. So the body has to burn ATP to move substances in active transport, just like your car burns gasoline essentially to move it when it goes down the road. The reason that energy has to be expended with active transport is we get substances that are moving from an area of lower concentration to an area of higher concentration. So if we go back to the example I used before where we've got 80 ions of sodium that are outside the cell and 1 ion of sodium inside the cell, in which direction would sodium move with active transport? It would move out from its area of lower concentration to its area of higher concentration. And because natural processes don't drive something to move from a lower concentration to a higher, for reasons that we're going to talk about with the next slide, this requires us to, in our bodies, use ATP and burn ATP, break it down essentially, to power the pumping of that particular ion against its concentration gradient. Okay, or that's another way of saying from its area of lower concentration to its area of higher concentration.
So this would only be applied to things that are big enough that it would have to go through an active transport. Otherwise, wouldn't it just passively diffuse back out?
It would. So you would have to have something that's complex enough, like a cell, that it could be using energy to power it. If you just had chemicals in a solution, active transport would never occur because it's not complex enough. You don't have energy being generated. You don't have pumps there that could pump it. So yeah. All right. So we have kind of talked about the differences between these two. The fact that energy is required, that we're moving from lower concentration to higher. No energy is required. We're moving from higher concentration to lower in these particular examples. What I want to do next is look at some specific ways that passive transport can occur, and then we'll look at some specific ways that active transport can occur as well.
So in active transport, would it always be using those proteins to pump it out?
It would. It would be using a protein pump to pump it out or in based on where the area of higher concentration is.
And for passive, does it sometimes still use that protein to come through, or is that going to be something that
It could. So if it was like the passive movement of sodium, obviously sodium can't just move through the membrane, so it would have to pass through a channel or a protein or something like that. And we're going to look at some specific examples of that. All right. But before we do, I want to kind of establish this idea. Maybe some of you are wondering, how the heck does something move without us expending energy from one area to another? When it's moving down its concentration gradient, or when it's moving from an area of higher concentration to an area of lower concentration. So I want to draw a picture for you that maybe hopefully will help to illustrate this a little bit. Here is a beaker that's filled with water. And if we added a drop of dye to the beaker, what happens over time? Okay, hey, it disperses, right? So originally we end up with this area where the drop of dye was released into the beaker that's very, very dark. But if you just let it sit there for 12 hours, you're going to come back and that dye has dispersed itself everywhere essentially in the beaker until it's spread out. And you get kind of this uniform lighter blue color since we're using a blue dye. Well, here's what's actually happening, okay, in that drop of dye. So here's my drop of dye. If you can imagine looking at it with a very high-powered microscope so that you're actually seeing the dye molecules within it, what you would see is there's all these little individual dye molecules, okay, and in that little drop, they're very highly concentrated, and they are always moving, okay? So there's this process which is known as Brownian movement. And what that means is molecules aren't sitting there stagnant. They're not sitting there just in one location. Even in our bodies, even in tables, even in things that seem to be solid, those molecules are actually moving. Okay, so we've got all these molecules that are moving around in this drop of dye, and as they're moving around randomly, what's going to be happening to them when they meet each other? Okay, they're going to have collisions. Okay, and so this guy is going to be moving around and he's going to collide, and when he collides, that's going to push him away from that other dye molecule. This guy's going to be moving around and he's going to collide, and when he collides, that's going to push him away from the other dye molecule. Okay, and we've got all these collisions that are occurring. What's going to tend to happen to these molecules of dye as they collide into one another and then basically bounce off each other in opposite directions? They're going to spread, okay? They are going to spread until we go from highly, highly concentrated molecules of dye to molecules that are spread out and they are uniform in their spread. Hopefully that looks uniform, okay? So we're going to go from this situation where they're having collisions, they move off in opposite directions, and that's going to tend to spread them uniformly throughout the container, okay? Now, once we get to this situation, they're still moving, but they're not having nearly as many collisions, okay? So there's not something that's tending to drive them in one direction or another. This is what leads to the movement of substances down a concentration gradient, okay, or from an area of higher concentration to an area of lower concentration. The same thing would happen if you put a sugar cube in water, essentially, and that's represented by these pictures with the beaker. You put it in, okay, it's a solid. Water starts to break those chemical bonds. Molecules are moving, they're crashing into each other. As they're moving and crashing into each other, they're coming off in opposite directions. That's tending to move, okay, the sugar molecules from areas of their higher concentration to areas of their lower concentration. If you look at this diagram here, it's still highly concentrated down here. We're going to be having more collisions in this area. That's going to move things away from that area. We're going to be having less collisions up in here, which is going to tend to not move things as much or at as high of a rate from this area until we establish this situation here. This situation where we've got an equal concentration of the molecules in all areas is what is known as equilibrium, okay? And equilibrium is a result of passive transport because things just tend to spread until you no longer have an area of higher concentration and an area of lower concentration. Anything about this that I could explain better? Okay. There's one other thing that I want to say about this that is kind of key to some things that we're going to talk about later. And that is, as you have these molecules colliding into each other and then bouncing off in opposite directions, you've got some kinetic energy, and you've got some energy of movement that's occurring. And actually, as things spread themselves down their concentration gradient, as they move from an area of higher concentration to an area of lower concentration, that is going to release energy that the cell can capture to perform work, okay? So we're going to talk about that, probably not today, probably the next time we meet. But I just want to say that while we're on this particular process and you've got this idea of movement in your head, that the cell can capture the energy from that movement and use it to power things. So we've kind of established what passive transport is. We've talked a little bit about why it occurs through Brownian movement. What I want to do now is give you probably 2 or 3 examples of actual types of passive transport that occur in the human body and give you a chance to think about them and think about how they work. So the first of these is a process which is known as simple diffusion. And what happens in simple diffusion is you need to have a concentration gradient. Okay, you've got to have an area of higher concentration and an area of lower concentration to drive the diffusion process. You have to have a cell membrane that is permeable to the substance that is trying to diffuse. And when you meet those 2 criteria, you can actually see that substance just moving from its area of higher concentration to its area of lower concentration across the cell membrane. So I've got a couple of examples here to maybe help you think about this, visualize this a little bit more. We've got a cell, okay? Each one of these little pink circles that I've drawn represents a molecule of carbon dioxide. So we need to first establish that the 2 necessary things for passive transport for simple diffusion to occur are met. Is the cell membrane permeable to carbon dioxide? It is, right? That's one of those very small fat-soluble things that can pass. Do we have a concentration gradient with this cell, or an area of high concentration, an area of lower concentration? Okay, where is our concentration highest? Inside the cell. So if we know with simple diffusion it's a passive transport process, the substance moves from its area of higher concentration to its area of lower concentration, or down the concentration gradient. In which of these areas are we going to have more collisions? Okay, we're going to be having more collisions inside, and as we do, that's going to tend to push things in opposite directions, and that's going to tend to drive this particular molecule outside of the cell. So from its area of higher concentration to its area of lower concentration. We look at this cell over here, okay, still talking about the same process, simple diffusion, where a molecule is just moving across the cell membrane. It's just squeezing between adjacent phospholipids because it's small enough to do so, and it's hydrophobic. Again, looking at carbon dioxide, so that's something that's permeable. It can cross through the membrane. We've got a concentration gradient, an area of higher concentration, an area of lower concentration. If this situation is met in the cell, where are we going to be having more collisions? Okay, outside the cell, right? Which is going to tend to push things into the cell. Things are going to tend to disperse from their area of higher concentration on the outside to their area of lower concentration on the inside. Okay, so just one example of how passive transport and specifically simple diffusion actually works in the body.
So would that happen when you're holding your breath then? Since
it's happening all the time.
Build up?
It's happening all the time. So yeah, so you're changing concentration levels there.
Like I understand the diffusion, but why would CO2 want to go back into the cell?
So you're not going to holding your breath. You're going to pass out before you have more carbon dioxide in the blood than there is in the cell. So you're changing levels a little bit, but it's not like you're changing that concentration. Now, if you did somehow change the concentration to where it was this, there was more carbon dioxide outside than in, you'd be dead. But it would start to move into the cell.
So that's not a very common occurrence compared
No,
to
this is just illustrating a point of how it happens. So I'm not saying that this occurs or this occurs. This actually does occur.
Okay.
It's just illustrating how this particular process works. All right, so that's simple diffusion. Here is another type of passive process that can be used to move something across the cell membrane without having to expend energy or ATP to actually pump it. This is what is known as facilitated diffusion. So a lot of times, don't make this mistake, a lot of times I've noticed that students see the word facilitated and then they say, oh, something is helping it because it's facilitated, and what's helping it must be energy, and that must mean that this is an active transport process where we're expending energy to move something. It is not. Okay, this is still a diffusion process. What happens in facilitated diffusion is you are moving something that the cell membrane is not permeable to. So it is something that can't just squeeze between the phospholipids because it can't be in the hydrophobic environment that we have within there. So what we've got instead are channels, okay, or transport proteins, thousands of them that are embedded in the cell membrane that allow the substance to cross. but it's still moving from its area of higher concentration to an area of lower concentration, and so it's still passive transport with no energy being required. If you look at this diagram here, here's kind of a good example of this. We've got a segment of a cell membrane, our phospholipid bilayer. We've got some channels or proteins that are embedded in it. You'll notice that inside the cell, the artist is saying this is our region of lower concentration. Outside the cell, we've got a region of higher concentration of some molecule, okay, some molecule that's represented in gray. And so, of course, we're going to be having more collisions out here, which are going to tend to drive things to this area here where we're having less of the collisions, essentially. The kicker is these things have to be driven through a channel or through a protein. So I had a student ask me one time, so this is like playing pool and you're hoping to like shoot it into the right pocket and it crosses the cell. Not so much, because there would be tens of thousands of these proteins embedded on the outside of the cell. So it's not like you're just trying to hit this one protein and it's very difficult and this process isn't occurring very rapidly. It can still happen very rapidly because there are so many of the proteins, the channels, actually embedded in the cell membrane that would allow, okay, this molecule on the outside to pass and cross into the cell where it's at a lower concentration. Yeah?
Wouldn't it cause ATP in order for it to pivot like this, or is it just like some sort of automatic process that causes it to pivot so that it comes through?
So it could cause ATP to open it initially, but if you think about once it's open, we're not directly using ATP to move it across, and that's why this is a passive transport, because we can use ATP just to open it, and then we can have all we want coming through. So the ATP is not being expended in the actual movement of the molecule, it's being expended in something that happened previous to that to allow it to move. And so the process of transport itself is not requiring energy. All right. Here is one last passive process. This is a process which is known as osmosis. Osmosis essentially is the diffusion of water. Okay? When we talk about osmosis in this class, we're always going to be talking about water actually diffusing down its concentration gradient or from its area of higher concentration to its area of lower concentration across the cell membrane. So there's a couple of things that have to occur for water to cross the cell membrane itself. The first thing is represented, or I'm trying to represent in this example here. I've got a cell. You'll notice we have a concentration gradient. Okay, so we've got more of some molecule on the inside, less of that molecule on the outside. But, okay, whatever this molecule is, the cell membrane is not permeable to it. Okay, and there is not a channel or a transport protein for it. So there is absolutely no way for what's on the inside of the cell to cross to the outside of the cell, to move down its concentration gradient, to move from its area of higher concentration to its area of lower concentration to establish equilibrium. Okay? Even when this is the case, equilibrium still tries to be established so that we have an equal concentration on the two sides of the membrane. If you think about a cell, it's filled with water, it's bathed with water on the outside. What can we do to establish equilibrium, to water this down essentially so that we've got an equal concentration on the two sides of the membrane? Okay, we're going to move water. In which direction are we going to move it? We're going to move it inside. Okay? If you think about this, we've got a higher concentration of our molecule, which means a lower concentration of water on the inside. We've got a higher concentration of water, lower concentration of our molecule on the outside. The molecule can't move, okay? It cannot diffuse, there's not a channel, there's not a transport protein for it. So the only way that equilibrium could be established in this case is for water to move from the outside in to basically water down the high concentration of solutes in here and establish a situation where we've got now an equal concentration basically on the two sides of the membrane. No ATP is required in this process. Water is moving down its concentration gradient from an area of its higher concentration to an area of its lower concentration. We're still establishing equilibrium through this particular process, and we only see osmosis happening when there's a concentration gradient to drive it and the membrane is impermeable to our solute, our molecule that basically is dissolved on the inside of the cell. So we've got about 5 minutes left, and that'll give us just enough time to work through this activity that kind of helps you to understand, think about this process of osmosis and what's driving it and how it actually occurs. If you look at these pictures that I have down here, I've got 3 pictures of red blood cells. So here is a normal red blood cell that you can see here. You can see kind of that concavity in its shape, which allows it to be very flexible and gives it that large surface area to volume ratio. What do you think has happened to this red blood cell? It's lost water. Okay, so water has left it and it's shriveled up. And if you look at this red blood cell here, what's happened to this guy? It's gained water. Okay, so we have caused osmosis to occur with this cell and osmosis to occur with this cell. And of course, if that happens, you're changing the shape of the cell. This cell is not going to function right because its shape is screwy, and this cell is not going to function right as a red blood cell either because its shape is screwy. What we can do to change the shape of a red blood cell or any cell of the body is place them in particular types of solutions. Okay? So what we're going to be looking at with this activity is what's known as tonicity. Tonicity is the ability of a solution, a liquid with something dissolved in it basically, to change the shape of cells by altering their internal water volume through osmosis. And we've seen this occur to these red blood cells, that they've lost water or they've gained water because of the solution that we've put them in. So just 3 examples for you. If we put a red blood cell in what's known as a hypertonic solution, this is water basically that has a higher concentration of non-penetrating solutes dissolved in it than what we actually see in the sea or in the cell. Okay? Seawater's a good example of that. So if that abstract definition didn't do anything for you, here's a visual representation. Here's a red blood cell. Okay? Red blood cells have a certain concentration of salt, a certain concentration of sodium ions within them. If we take a red blood cell and we put it into salt water, okay, there is much more sodium in the salt water than there is in the red blood cell. So if we answer these questions here, where's our sodium concentration higher? Outside the cell. If the membrane is impermeable to sodium, we're just going to go with the assumption that it is so we can kind of illustrate how osmosis works. Which direction will water move? It's going to move out to try and establish equilibrium on the 2 sides of the cell membrane. And what's going to happen to our cell? Okay, it's going to shrivel like the one that we saw in the previous picture. So here's another example here. We've got a red blood cell that's been placed in a hypotonic solution. So hypotonic would be a solution where there's less solute dissolved in it than what we actually see in the cell. So here's kind of a visual representation of that. We've got more sodium inside the cell than we have outside the cell. So we do have a concentration gradient, and it's higher inside the cell. If the membrane's impermeable to sodium, in which direction is water going to move? It's going to move in, and what's going to happen to our cell? It's going to expand, right? It's going to fill with water, and that's going to completely deflate what the cell is able to do functionally because its shape is no longer correct. It doesn't have the correct form. Here's one more. So an isotonic solution is a solution that has the same concentration of solutes as is found in the cell. So here's my visual representation of that. What I'm intending to show is that we've got the same concentration of solutes on the 2 sides of the membrane. So we don't have a concentration gradient, right? We don't have an area of higher concentration and an area of lower concentration. If the membrane is impermeable to sodium, in which direction will water move? Okay, everything's going to stay the same. So we're not going to have a net movement of water here or a net movement of water there. What's actually going to happen is it's going to move in both directions equally, okay? But if that confuses you, we can just say it doesn't move. It doesn't have to move because we've already got equilibrium established essentially. What will happen to the cell in that case? It's going to stay the same shape, same size, be able to function. This is why when somebody's given an IV, it is 0.9% saline. Okay? If you gave them a saltwater IV, what would happen to their red blood cells? They would shrivel. Okay, if you gave them a distilled water which has no solutes in that IV, what would happen to their red blood cells? They would expand. They would take on water. So very, very important that you get a 0.9% saline IV, because 0.9% is the concentration that's in your red blood cells. And anything above that, anything below that is going to change the shape of those cells and prevent them from being able to function, and you will die.